Okay, good morning, everyone. It's both an honor and a privilege to introduce our speaker today, Dr. Doug Lowy. Doug received his at NYU, and then I'm proud to say received his medical residency at Stanford, subsequently trained in dermatology at Yale. He then moved to the NCI as a senior investigator, and since that time, he's held a number of high-level positions, including as acting director from 2015 to 2017, and again in 2019. He's currently the principal deputy director at the NCI and chief of the laboratory Cellular. Together on Schiller, Dr. Lowy's made many truly groundbreaking contributions to our understanding of the viral pathogenesis of malignancies, with particular reference to the human papillomavirus. Their work has elucidated the oncogenes encoded by the virus and culminated in the development and clinical testing of 3 highly effective HPV vaccines that are now in clinical use worldwide. It's also notable that the work has major implications for the prevention of cervical cancer, among other cancers, which remains rampant and a leading cause of death of women in many low and middle income countries. He's also studied members of the RAS gene family with recent work focusing on the tumor suppressor activity of DLC1, a GTPase activating protein that negatively regulates the Rho family of GTPases. If this were, Doug has also led the SARS-CoV-2 serology research at the NCI. He's received a large number of awards that I can't go into in great detail, but they include, among many others, the Albert Sabin Gold Medal Award, the Harrington Prize from the ASCI, the St. George Prize, and in 2017, the Lasker DeBakey Clinical Medical Research Award. I would note that this Friday, March 4th, is International Awareness Day, so it's particularly relevant that we have this talk on preventing cervical cancer and other HPV-associated cancers by vaccination and screening. Doug, thanks so much for joining us.
Beverly, good morning to you and everyone online. Thanks so much for this lovely introduction. And I also want to thank Stanford for the invitation. I'm only sorry that we can't be there in person because I would love to be able to interact with you as well as many other colleagues at Stanford. Maria, if you could have the first slide, please. I'm really going to divide my presentation into sort of 2.5 parts. The one half is I'm going to talk a bit about the epidemiology of HPV-associated cancer. Then I'm going to discuss the HPV vaccine. And then in the last few minutes, I will discuss screening, and not just cervical cancer screening, but some very new results that make it likely that in the not too distant future, there may be standard of care screening, at least for high-risk patients for anal cancer. So if I could have the next slide, please. This is my disclosure slide. And I am an inventor of the FDA-approved vaccines, as Beverly has mentioned. But the really important part of this slide is in green. I will discuss a potential off-label use of the vaccines, which is fewer vaccine doses, that is 1 dose. I want to make it clear from the outset, this is not standard of care in the United States, and I am not advocating for it to be standard of care in the United States, but there is a lot going on in research in this area. Next slide, please. So in terms of the epidemiology of HPV-associated cancers, the next slide will show you that it really depends on where you live. If you'll look on the next slide on the left side, this is taken from a review a few years ago from IARC about the incidence and mortality of cervical cancer in different parts of the world. In blue is the incidence, and in red is the mortality. And the third from the bottom is Northern America, which is the United States. And what I hope you can appreciate is that we have both a low incidence and a low mortality rate from cervical cancer. But if you go up to the upper areas of the slide, not only are the blue areas much bigger, but the relationship between the blue and the red is also more closer to 1-to-1, because a higher percentage of women who develop cervical cancer, particularly in Africa, are destined to die from it compared to high income countries. Now, if you turn your attention to the right side of the slide, low and middle income countries, 90% of cervical cancer incidence and death occurs there. And most important, the World Health Organization projects that the incidence and mortality will increase by about 40% every 15 years with current approaches. And in low and middle income countries, cervical cancer represents about 90% of HPV-associated cancer, and it affects about 95% of HPV-associated cancer affects women. The United States was a high incidence country in the 1950s, but Pap smear testing and treatment of cervical precancer enabled us to go from being a high incidence country to the low incidence and mortality country that we are now. Next slide. So we're very fortunate that when it comes to HPV-associated cancer, there's not one intervention, but two that can dramatically decrease the risk of cancer. Vaccination can prevent the development, as far as we know, of the vast majority of HPV-associated cancer. Screening, on the other hand, is approved just for cervical cancer screening, which has been very important, and as I will point out, continues to be critically important. It is persistent chronic HPV infection with a oncogenic HPV type that increases the likelihood of women progressing to invasive cancer. And the pathogenesis seems to be quite similar for the other HPV associated cancers. Next slide. So this slide summarizes for you the three different HPV vaccines that are FDA approved. Let's take them in order from top to bottom. Cervarix, made by GlaxoSmithKline, is virus-like particles composed of HPV-16 particles and HPV-18 particles, because as you'll see on the next slide, they account for about 70% of cervical cancer and an even higher percentage of the non-cervical cancers. The adjuvant that's used in Cervarix is stronger than the adjuvant used in Gardasil and Gardasil 9. So there actually is some cross protection against some of the HPV types present in Gardasil 9. But Gardasil, made by Merck, in addition to particles from 16 and 18, also has particles from HPV 6 and 11, which are responsible for about 90% of genital warts. Gardasil 9, in addition to having the particles from Gardasil, has particles from the 5 next most oncogenic types. And Gardasil 9 is the only HPV vaccine that is currently available in the United States, although the other two vaccines are FDA approved. Next slide. So this slide summarizes for you the different HPV-associated cancers and their relative frequency in the United States. You can see that there are about 35,000 cases per year in the US. And the darker blue is those caused by HPV 16 and 18. The lighter blue is those caused by the additional 5 types in Gardasil 9. And the very light one is caused by HPV types that are not in the any of the vaccines. What I hope you can appreciate is that the incidence of oropharynx cancer actually is now higher than the incidence of cervical cancer. And oropharynx cancer represents, is much more common in males than in females. About 75% of those cancers occur in men and about 25% in women. And so the take-home message is that probably around 90% of HPV-associated cancer can be prevented by the current HPV vaccines, especially Gardasil 9. Next slide. And I am interspersing some of the presentation with some comparisons between HPV and SARS-CoV-2 because it has been so prominent over the last 2 years. First, this slide talks about the disease and the viruses. The disease caused by HPV, there are several HPV types, as I mentioned on the last 2 slides. On the right, with SARS-CoV-2, disease is caused just by a single type. On the left, HPV is a DNA virus, and the implications there are that the viral genome rarely mutates. On the right, however, SARS-CoV-2 is an RNA virus, and it commonly mutates. And then on the left, viral variants within a single HPV types are closely related immunologically, where, as we have seen very recently with the Omicron variant, viral variants for SARS-CoV-2 may be distantly related immunologically. Next slide. So this slide is just a summary about the safety of the HPV vaccine. This was published in 2016, 10 years after the first HPV vaccine was FDA approved. And I've highlighted in this abstract, this is from the CDC, safety data have been reassuring with no confirmed safety signals identified. Next slide. These are 3 of the initial HPV vaccine results. First, the initial studies used 3 doses over 6 months. And the reason was there was no precedent for an effective vaccine against a local sexually transmitted disease, which is what HPV infection is. Second, the vaccine has very high efficacy, greater than 95% against the HPV types in the vaccines. In principle, it can protect against the vast majority of cancers attributable to HPV infection. And then the third point is that the vaccine confers sterilizing immunity in most vaccinees. That is, it prevents infection in most vaccinees. Next slide, please. This slide actually shows you data from that were published a year ago from Denmark, showing indeed that the HPV vaccine can prevent about 90% of cervical cancer. Let's look first on the graph on the left. In the black is shown the incidence in young women of cervical cancer in Denmark. These are the women who were unvaccinated. And then in the red is shown the incidence of cervical cancer in women in Denmark who were vaccinated. And there's almost a 90% difference. If you now turn your attention to the right side, the rightmost graph, this is women who were vaccinated when they were 20 to 30 years of age, and there's no clear protection. The one on the left was women who were vaccinated when they were 16 or younger. And in the right, as you would imagine, the women who were vaccinated when they were 17 to 19 years of age, there is some protection, but nowhere near as much. And what this is telling us is, as shown in the clinical trials, HPV vaccination is very effective at preventing new infection, but it does not alter the natural history of established infection. Next slide. So when it comes to the uptake of the HPV vaccine in the United States, we were not a high adopter in terms of uptake up until relatively recently. And the CDC has had a fairly successful campaign And one of their public service announcements is this slide that I really love for those of us who may be mathematically challenged, where one vaccine, 2 doses equals protection against 6 types of cancer. Next slide shows you what has happened over the last 10 years in terms of the uptake of the HPV vaccine among boys and girls who are 13 to 17 years of age, who have had 1 or more doses. If you look at 2010, there's this tremendous imbalance between the percentage of girls who were vaccinated versus boys. That's because it was only in 2011 that the FDA made a so-called routine or strong recommendation for the HPV vaccine for boys. If you now fast forward to 2015, the percentage of girls who were vaccinated was now a little over 60%, but boys have really come up to be 50%. And if you now fast forward to 2020, which are the last year for which we have data, and although it was most of that year was part of the pandemic, the uptake for girls is over 70%, and the uptake for boys is also over 70%. And it seems likely that we are going to get to close to 80% in another few years. Next slide. So this slide is taken from Kaiser Permanente in Oregon, and they're, basically the patients that they follow. And if you look on the left, the topmost line, the black non-interrupted line, the vaccine recommendation was in 2006, and this looks at genital wart incidence. And what I hope you can see is that there's a really dramatic decrease in the incidence of genital warts starting in 2007 and continuing through 2016. No such change with chlamydia or gonorrhea, 2 other sexually transmitted infections for which we don't have preventive intervention. If you look on the right, for males, the strong vaccine recommendation in 2011. And once again, after 2011, a substantial decrease in the incidence of genital warts. Next slide. This slide shows you what has happened to an uncommon but serious complication of women who are infected during pregnancy and delivery. It's the development of juvenile onset recurrent respiratory papillomatosis. And the children, and what I hope you can see is that the vaccine was approved in 2006. And in the last 3 periods that were looked at, the incidence of respiratory papillomatosis has gone down substantially, regardless of the criteria that are used. And this is really quite impressive. Next slide. This is in the United States. So what about duration of protection? I'm not showing you the data because they are published, but one we found in our Costa Rica vaccine trial, which was started before the FDA approval of the vaccines, that the GSK vaccine, Cervarix, 1 vaccine dose prevented HPV-16 and 18 infection as effectively as 2 or 3 doses 1 year after vaccination. This is post hoc analysis, not sufficient to change standard of care. 100% of the women who received 1 vaccine dose remained positive for HPV-16 antibodies 11 years after vaccination. And the data for these statements are in this paper that was published in 2020. Next slide. What about in the United States with the development of herd immunity? This is a complicated slide, but I think it's worth your effort to look at it. If you look on the left, what I have bracketed in the open red rectangle is the prevalence of the HPV vaccine types in unvaccinated women who are 20 to 24 in 2018. And what I hope you can see is that in 2007 to 10, it was high. Then in orange in 2011 to 14, it went down. And then in purple in 2015 to 18, it was down further. And this is strong evidence of herd immunity, because this is happening in the unvaccinated population. If you now look on the right in the red open rectangle, this is in women who were 5 years older than the 20 to 24-year-old cohort, and there's no such stepwise decrease. So in these women who are 25 to 29, there is no herd immunity yet. But we can imagine that as the 20 to 24-year-olds age and become 25 to 29-year-olds in a very short time period, that there will be herd immunity among 25 to 29-year-olds. Next slide. So this slide is another comparison between the HPV, now between the vaccine and the SARS-CoV-2 mRNA vaccine. Vaccination with HPV prevents even benign infection in most vaccinees. SARS-CoV-2, many vaccinees remain susceptible to benign infection, especially as we saw with Omicron. On the left, HPV vaccination readily induces herd immunity, but with SARS-CoV-2 vaccination has not yet been shown to induce herd immunity. The next slide shows two additional comparisons. Next slide, please. And the HPV vaccine confers strong protection against all variants within a given HPV type. But as we saw with Omicron, the SARS-CoV-2 vaccine may confer reduced protection against some variants. In addition, the HPV vaccine induces more than 10 years of strong protection, at least in post-hoc studies, whereas the SARS-CoV-2 vaccine thus far has been shown only to induce several months of protection. Next slide. So I now have a few slides to try to look at, well, what might be the explanation for the durability and effectiveness of the vaccine? We think that the secret sauce is the repetitive structure of the virus-like particle vaccine. And it induces high levels of durable neutralizing antibodies that confer long-term protection against HPV infection and disease. And on the right in red are shown neutralizing antibodies, and on the left in the blue are particles without the antibodies. Next slide. A number of years ago, Patricia Day, Rhonda Kynes, and their colleagues developed a mouse challenge model. And diagrammatically, this is a genital tract challenge model, and this is using HPV-16. In immunized mice, the dermis contains a lot of antibodies, but in the intact mucosa, there's not much. But in order to get infection, you need to abrogate epithelial integrity. And that's shown diagrammatically with the next click. And that then leads to, as shown in the next click, that the antibodies go into the area of potential infection. Papillomaviruses have not read virology journals. And so they don't know that the first step in every virus infection is to bind to the cognate receptor of the cell. Instead, papillomaviruses actually bind to the basement membrane. That's their first step. And only later do they then bind to the basal cell. But the presence of the antibodies, as shown in the next click, when the virus comes in, as shown in the next click, the antibodies bind to the virus and they prevent, as shown in the next click, the binding to the basement membrane, and so you don't get infection. Next slide. So we think that this is attributable to the dense repetitive epitope array of the virus-like particles. And on the left is what is done with many vaccines. They are monomeric protein complexes. And as a result, there are weak activation signals, a low level of antibody, and they're of short duration. By contrast, the virus-like particles get strong survival proliferation signals, high levels of antibodies of long duration. So really, we think that the key to this is the oligomerization of the B cell receptor by the VLP antigen that is induced in long-lived plasma cells. Next slide. So we've spent a fair amount of time in the last few years trying to evaluate whether a single HPV vaccine dose might confer years of protection. The next slide gives you the rationale for it. The need is that HPV vaccination of more than 40 million women in each birth cohort will be needed if we're going to really deal with the worldwide problem of cervical cancer. Most of these women live in low and middle income countries. And just for your denominator, each birth cohort is about 60 million women. The current reality is that only about 10% of eligible young women in low and middle income countries get vaccinated each year, 15 years after FDA approval of the first vaccine. We think that a possible solution could be single dose HPV vaccination. It would be less expensive and logistically easier than 2 doses. But as I've said more than once, it's not yet standard of care. Next slide. So Amy Kramer from NCI and Rolando Herrero from Costa Rica are doing the Costa Rica 1-dose HPV vaccine trial. This is a large non-inferiority efficacy trial to compare the efficacy of 1 dose versus 2 doses of 2 FDA-approved vaccines, Gardasil 9 and Cervarix. We don't expect the data from this trial to read out for another year and a half or 2 years, in part because of COVID. But I can tell you that based on the design of this trial, the Bill and Melinda Gates Foundation supported the development of a faster trial. Next slide. And that trial is known as the Kenshi trial, which Ron Barnabas from the University of Washington is the principal investigator. And this is in Kenya, where it's a 3-arm trial of 15 to 20-year-old women. All women in the trial received 1 dose of a vaccine. 1 arm was Gardasil 9, a 2nd arm was Cervarix, and the 3rd arm was meningococcal vaccine. The results were recently reported at the International Papillomavirus Conference, and the results showed a 97% vaccine efficacy at 18 months against new persistent HPV-16 or 18 infection from a single dose of Gardasil or Cervarix. So next slide. And less than a month ago, the United Kingdom Joint Committee on Vaccination and Immunization, the JCVI, it's a bit like the Advisory Committee on Immunization Practices in the United States, recommended a single dose for the HPV vaccine for the United Kingdom. That has not yet been adopted by the United Kingdom. It probably will be several months before the UK makes a decision whether they will adopt this recommendation. Next slide. So I'm now going to turn for a few minutes to discuss cancer screening. First, cervical cancer screening. And the take-home message is that molecular testing is replacing the pap smear, or as one of my bad jokes is, this is not your mother's cervical cancer screening. Next slide. We've had a substantial decrease in the incidence of cervical cancer over the last years. Back in 1975, the incidence, if you look on the left, of cervical cancer among Black women was about twice as high as the incidence among white women. The Black women are shown in the blue triangles and the white women in the green triangles. Over the years, the incidence has really come down substantially faster for Black women than for white women. So now the incidence is quite similar. However, mortality rate still lags substantially, especially for Black women whose mortality rate is about 50% higher than that of whites. And you can see that Hispanics and Native Americans are kind of in between. This is SEER data. We are trying to work with the FDA to see if it's possible to get self-collected vaginal specimens for cervical cancer screening approved, because this could enable the screening of hard-to-reach women, which could decrease cervical cancer incidence and mortality. Next slide. And one of the important reasons that I am talking about screening in addition to vaccination is that Emily Berger and her colleagues from the Harvard School of Public Health have recently done a modeling comparison of what happens if you were to scale up screening to 90% coverage in the United States versus scaling up vaccination to 90% for girls only. And what their modeling shows is that you could get down to 4 cases per 100,000 10 years earlier if you scaled up screening compared to scaling up vaccination. So it's critically important that we be able to screen women who not only have been being screened regularly, but also hard to screen women. And because of COVID, we know that many, not just women, but cancer screening in general has lagged because of COVID. And if COVID is winding down or becoming more endemic, getting back to regular screening, the importance cannot be emphasized too greatly. Next slide. So what about screening? HPV testing is replacing cytology for primary cervical cancer screening. And the reason is that it's more sensitive and enables longer intervals between screens. However, many HPV positive women do not need to be treated because they do not have precancer. Many HPV infections will go away without treatment before the lesions become precancerous. And therefore, ancillary testing, in addition to HPV testing, has become important. And for triage of HPV positive women, there are various triage tests. One was 2 years ago approved by the FDA, which is using P16 and Ki67. P16 is the tumor suppressor gene that in most cancers, the expression is decreased. But paradoxically, in HPV-associated cancer, P16 expression is actually increased, and this is because of the effects of the E7 protein of on P16. There are still experimental studies looking at viral DNA methylation or viral E6 protein expression. The next slide shows you some data from Nico Van Vensen and Mark Schiffman at NCI, who have really pioneered this approach, the dual stain positivity of P16 and Ki67. And this is taken from a paper from a little over 5 years ago, where the risk of either CIN2 or CIN3, if you are dual stain positive, the dotted horizontal line, the black one, is the threshold for colposcopy. You should go to colposcopy. If you're negative, you don't need to go to colposcopy. So it really is a pretty dramatic test. Next slide. And this is the last slide on cervical cancer screening. 2 years ago, the American Cancer Society recommended substantial changes in cervical cancer screening. First, to begin screening when women are 25 years old instead of 21. Herd immunity in the United States from HPV vaccination enables screening guidelines to be the same for vaccinated and non-vaccinated women. They prefer HPV-based screening, although cytology is also standard of care because it can be done every 5 years if a woman is negative. And the management is risk-based. It's based on molecular biomarkers, not based on cytology. The United States Prevention Services Task Force recommendations have not changed. It remains to be seen whether they will. The last topic that I want to discuss, next slide, is anal cancer. And this, again, is not yet standard of care, but there recently, the code was broken on a long-term trial supported by the NCI, and the results were positive. They were presented last month at a conference, so I want to show this to you. First, the epidemiology. Next slide. This is the SEER data, and although HIV positive men are at very high risk of developing anal cancer, when you look on a population-wide basis, women are more likely to have anal cancer than men. And the incidence of anal cancer, both in men and women, has been increasing since 2000, with the exception of the last couple of years for women. And this is probably because the incidence of HPV infection going up and there's no screening. And it's too early for vaccination to have had an impact on this area. Next slide. So the anchor trial, anal cancer HSIL outcome research study, which is high-grade dysplasia, the hypothesis that was tested was the treatment of anal HSIL in high-risk patients can reduce the risk of anal cancer. It's a multi-center randomized controlled trial in HIV-positive men and women. It was launched in 2015 and NCI sponsored. Joel Palevsky from UC San Francisco is the principal investigator. And in the last part of last year, the Data Safety and Monitoring Board for the trial stopped it early because it had achieved its efficacy endpoint after the development of 30 anal cancers. The results were presented last month at the Conference on Retroviruses and Opportunistic Infections. Next slide. So this was over 10,000 men and women who were living with AIDS, and about half of them had biopsy-proven anal high-grade dysplasia. During the time, there were 17 individuals who were diagnosed with anal cancer. Next slide. And this slide, which I think is really the take-home message, is the men and women who were treated for high-grade dysplasia are shown in red, and their cumulative incidence of anal cancer. And the men and women who were treated with active monitoring are shown in blue. And what I hope you can appreciate is that there was a more than 50% decrease in the risk of progression to anal cancer among the men and women who were treated for their high-grade dysplasia. This is not yet standard of care, and this is really just for high-risk people. But hopefully, there will be studies in the future that will look at lower risk people, especially since women, actually more women than men, develop anal cancer, and presumably the FDA will look at these data, and regulatory agencies may end up making this standard of care. But needless to say, not me. Next slide. So I'd like to summarize what I have been discussing. Basic research led to identification of HPV as the cause of several cancers. and to development of HPV vaccines, and to molecularly based cervical cancer screening. Virus-like particle display is highly immunogenic. It can induce herd immunity and durable protection. Most important, we think that the control of HPV-associated cancer as a worldwide public health problem may soon be feasible. And as I mentioned at the end, anal cancer screening for high-risk patients may become standard of care in the future. So thank you so much for the invitation. I hope that that's the last slide. Don't show the others. I really hope that I've left enough time for your comments and questions, which I would look forward to hearing. Thank you.
Thank you, Doug. That was really inspiring. I have to say, to make a difference like that across the world is an amazing feat. So I have some questions while we're waiting for questions from the audience. Having looked into developing HPV programs in some developing countries, low and middle income countries, the access to the vaccine remains a real problem. And it seems that the production is limiting. If you had any ideas about that, or whether there's some way to alter the production such that you can get more availability.
So there has been a shortage for the HPV vaccine. And that was noted, about 3 years ago. It's very unusual for a vaccine that is as old as the HPV vaccine to have a shortage. And the reason is several-fold. And I'm just speculating. First, people have been surprised that the HPV vaccine has tended to perform quite well, as I have shown you. Second, there has been a great desire for gender-neutral vaccination, so that this has led to an increase in the demand for vaccination. The expectation is Merck has committed to greatly expanding their production of the vaccine, and we anticipate that within 2 or 3 years, there will be plenty of vaccine available. In addition, there is regional vaccine manufacturing, both in China as well as in India and potentially other locations. So, and there is tiered pricing in low and middle income countries. Any company can manufacture and sell in low and middle income countries. So while there is this short-term shortage, and I don't mean to dismiss it, I think that the future looks really very bright for the availability.
Thank you. So we have some questions in the chat. Ron Levy asks, have VLP candidate vaccines been developed for COVID?
Yeah, well, the answer is to some degree, okay? We think that, if you will, the secret sauce is the repetitive structure. So, for example, the Walter Reed, people at Walter Reed are developing a vaccine that uses a ferritin base. And with the ferritin base, you end up with then 70 copies of the spike protein, okay, of the spike trimer, which is many, many more copies than what we imagine happens when you immunize with an mRNA vaccine or with the current vaccine platforms. It remains to be seen whether this will actually, in practice, lead to more, be more immunogenic, but more important, lead to more durable protection.
Thank you. Yes, thank you for a fabulous talk. Can you please comment on whether where we are with screening for oropharyngeal cancers?
So screening for oropharyngeal cancers is not yet FDA approved, okay? And the reason is that Maura Gillison and her colleagues, as well as others, have tried to look for whether doing some kind of HPV-based screening is useful. And at the moment, it wasn't. There is a experimental, if you will, experimental study where a number of studies first developed by Amy Kramer at NCI and others using antibodies to HPV-16E6. People can be antibody positive for years before they develop oral HPV cancer. But whether that approach will end up being standard of care, there's just much more research that needs to be done for that.
Let me ask as a follow-up to that, the intervention for the anal cancers that markedly decreased the progression, is that applicable to head and neck or other areas?
So I should have said that head and neck cancer, the big problem from a clinical perspective is that there is no clear pre-malignant lesion. That's the problem. You don't know what to treat. Too many people are HPV positive, have oral HPV positivity, even if you limited it to HPV-16. You can't go and willy-nilly, treat everybody. It would be a vast degree of over treatment. The anogenital infections all have pre-malignant lesions that can be seen and treated. That's the big difference, if you will, between HPV precancer below the waist and HPV precancer above the waist.
Thanks. A number of other questions. Becky Richards asks,
Hi, Becky.
Can you speak to the durability of sterilizing immunity after vaccination, even beyond the 11 years you discussed? Specifically wondering about your thoughts of moving HPV vaccines earlier in childhood as part of the early childhood vaccine schedule.
So I don't think that there are data other than, the prevalence of the way that the 2020 study was done was to look at the prevalence of HPV infection 11 years after the one dose. Okay, and so the prevalence was much, much lower rather than looking at cumulative incidence or something like that. But there are studies that have been done in 5 or 6 year olds with the vaccine, and people have discussed the possibility of doing a vaccination in infants or children who are 2 or under. The problem is that you'd be vaccinating people for a decade longer than currently vaccinating before you would see a benefit from the vaccine. So the target group now are young adolescents, and that's because it's very important to vaccinate before the adolescents become sexually active. But if you move it back another 10 years, it will take an additional 10 years. So that is, I think the durability is no longer going to be the big issue, but it really will be, what about the upfront investment?
Thank you. So Steve Arton asks, you showed that HPV vaccination induces sterilizing immunity and herd immunity, and that the mechanism may relate to blocking binding of the virus to the basement membrane. From your findings, can you make predictions on how to improve SARS-CoV-2 vaccination strategies to make them similarly sterilizing?
Well, I think that we should be putting more effort into trying to use repetitive structure virus display. Okay. People, it really is noteworthy how much longer the duration is. This is the HPV vaccine, is unusual for not being an attenuated live virus. Okay. It's just a subunit vaccine, and one dose seems to do a lot. And I think that trying to mimic it would not be unreasonable.
Thank you. Dan Kapp asks again about oropharyngeal cancer, about screening with serum antibody or saliva. Is that the current?
Yeah, so saliva testing has not tended to be as reproducible as as say blood. Okay, I mean, in principle, it may be that not enough effort has gone into saliva testing, but certainly antibody testing, perhaps in conjunction with something else, is something where I think is really ripe for research, because the problem with the vaccine is that it doesn't alter natural history. And the people who are going to develop these cancers are going to be in their 50s and 60s. So you're going to have this really long lag period before the impact of the vaccine will be seen in oropharynx cancer, and trying to develop interventions, whether they be screening interventions or therapeutic interventions that will be less debilitating than current treatment, are really highly worthwhile to consider.
And Melissa Bondi wants to bring up the vaccine hesitancy. Is the acceptance rate is there less hesitancy?
Hi, Melissa.
Also, internationally, will the dose be 1 or 2 doses as it becomes more available?
Yeah, well, vaccine hesitancy has been a problem, and it was dealt with quite effectively both in Denmark as well as in Ireland. Both countries experienced vaccine hesitancy at some time after the vaccine was introduced. And they ended up doing a multi-pronged approach, including having a publicized conference in Denmark and in Ireland, separate conferences. And it was, and this multi-pronged approach led to, if you will, increased uptake of the vaccine in both countries. The United States, the vaccine hesitancy has really been perhaps for somewhat different reasons. I think even Merck would agree that they shot themselves in the foot soon after the HPV vaccine was approved, because they tried to make it required in Texas and Michigan, both high-profile areas, and they didn't become standard of care. But the one thing that groups that had concerns because of the sexual nature of HPV infection was, they said, just please don't make the vaccine mandatory, okay? And so this led to a tremendous pushback, and even, ended up in 2011 as part of the Republican presidential primary debates. So it's a little bit different, but the vaccine hesitancy has been, if you will, every year there are a few more percentage points of people who are getting vaccinated in the United States, and I think the CDC, the cancer centers, the cancer centers have come out with a couple of unified statements. These are the only time that the cancer centers have all band together and come out with unified statements about the importance of HPV vaccination. All of this, no one thing makes the difference, but together they're really, I hope that my slide showed you how uptake has really changed.
Yeah, we're still not there in California, I'm afraid, but everybody continues to work on it. So Steve Artandi wants to know about the efficacy of checkpoint inhibitor therapy for HPV cancers.
Yeah, thanks, Steve. I think that checkpoint inhibitors should work, but they have had limited success thus far, and the reasons for that really remain to be elucidated.
So I think that's the end of the questions we have. I have to say, Doug, it's been a real pleasure to hear about the updates on this. We can't wait for the 1 dose data to come out so that we can implement it worldwide. That would be so exciting.
Well,
And,
I think, you know, if I may take my prerogative and editorialize, I think that the United Kingdom Advisory Committee taking the lead here is going to make it easier for low and middle income countries to move to 1 dose. Well, we have a 1 dose consortium that the Bill and Melinda Gates Foundation supports and is run by PATH. And the concern that was expressed last year during our meeting was that there could be a 2-tiered system. 1 dose is good enough for low and middle income countries, but not good enough for high income countries. So the recommendation from the United Kingdom, if the United Kingdom adopts it, I think really changes that dynamic substantially.
terrific. Well, once again, thank you so much. It's a pleasure having you talk to us, and we hope to get you here again soon. I know you were here last year, but love having you.
It was virtual. It was virtual also, Beverly.
Okay.
I just want you to know, I even have a jacket from the Stanford Cardiovascular Institute that I was planning to wear today when I would be on campus in Palo Alto, but maybe another time. Thanks so
well, we
much.
have to get you a Cancer Institute jacket.
I really
as well.
enjoyed being able to come, at least virtually. Bye-bye.
Thanks so much. Bye-bye.